developing an R package for annotating and visualizing 3C-based genomic data. So just a quick background. Certainly the use of chromosome conformation capture data or 3C-based data has been on the rise and gaining popularity, as you can see by the number of publications in PubMed referencing this method. And by combining this method with next-gen sequencing, we start to be able to capture chromatin interactions on the whole genome. But our group found that visualizing the data in this manner doesn't really give us a lot of information about the type of chromatin that's interacting. So we set out with a goal to develop software to aid in annotating and actually visualizing this data at the whole genome level. Does this thing work? All right. So to do this, we needed to integrate 3 different data sets. And the first is the chromatin interaction in data from something such as HiCAGE or ChIA-PET. The second is the chromatin annotations, which give us some information about the position of functional elements in the DNA, such as enhancers and promoters. And then lastly, RNA-seq data that gives us an idea about the level of expression of genes located near these functional elements. And I'll just make a quick mention of an additional package that our group has developed called StateHub, that for the data I'll show today, all of our chromatin annotations were generated using this framework for a fully reproducible chromatin annotation. However, you're free to use whatever chromatin annotations that you like. So our package, HiCAGE, integrates this data and produces a summary figure based on the Circo style plot, in which in the center of the plot we have, you can see the interaction between the different functional elements. And then surrounding those interactions, you can see in red the strength of the chromatin interaction, or the HiC score, and then surrounding that is the gene level expression of genes near those functional elements. Additionally, the package produces a upsetter style plot, and this is just a quantitative measure of the types of interactions that are occurring, such as the number of promoters and enhancers interacting, or promoters interacting with transcribed regions. And so the package uses processed data, but we can run thousands to millions of interactions and produce these plots in under a minute, which is pretty quick. So to validate this software, we turned to a recently published paper out of Richard Young's lab in MIT, in which that he showed, in contrast to CTCF, which we know binds CTF sites at the TAD domains, and forms these loops, that another similar protein, YY1, preferentially interacts with enhancers and promoters and forms loops of these elements. And so using this data from his paper, we fed it into our package, and what we find is that we could faithfully recapitulate what they found in that paper, in which on the left, the CTCF data was dominated by CTCF interactions as expected. And then in contrast, the YY1 data was largely dominated by promoter-promoter and promoter-enhancer interactions. So next, just to make it user-friendly, we developed a graphical user interface, and this is available online. So this produces the summary data frame of the annotated data, also the plots that you can download. And then lastly, we've started to incorporate gene ontology analysis, where we can actually start to explore Go terms that are associated with a specific interaction of interest, such as the Go terms associated with enhancers interacting with like a poised promoter. So a paper describing our work is available on bioRxiv and currently under review at Bioinformatics. The R package is available through GitHub, and lastly, the GUI is available on Junk DNA Lab. It's our local site, and it's a Shiny-based app. So just to thank the people that helped develop it. The people here bolded were really instrumental in the package development, especially my mentor Dennis and the rest of the group, and also the graduate program at Cedars-Sinai that helps support me. Thank you.